Hello, everyone. My name is Khristian Erich, and I'm an MD PhD student in the Longacre Lab. And I'm very excited to present my work on the role of creeping fat in intestinal fibrosis. So inflammatory bowel disease is a chronic illness that causes inflammation in the digestive tract. In a particular subtype of IBD called Crohn's disease, patients are susceptible to the formation of intestinal strictures. And you can see an intestinal stricture here in figure 1, where the normal bowel is replaced with scar tissue that accumulates over time and causes a bowel obstruction. It's estimated that Crohn's disease patients will present with strictures up to 50% of the time, and surgical intervention is often required in about 80% of these cases, and 70% of these patients will have some type of recurrence. And so as a result, intestinal strictures represent a significant clinical burden. And if we take some histology of these strictures, what you can see is the presence of a significant amount of collagen as well as muscle that replaces the normal intestinal epithelium. And this is what leads to the absorptive issues that these patients have. And so one of the interesting observations, as you can observe here in figure 1C, is that creeping fat, which is mesentery that wraps around the bowel in the context of Crohn's disease, is oftentimes very associated with the formation of strictures. And we wanted to ask the question, why? Is the mesentery simply a passive bystander, or is it actively involved in intestinal fibrosis? And so our hypothesis was that there may be a subset of adipocytes that are present in the creeping fat that are capable of converting into fibroblasts and thus promote fibrosis. And so to address this question, we needed to first develop a model of intestinal fibrosis and stricture formation. And so to do so, we utilized a surgical model whereby we created a colotomy in the bowel, and then we closed it transversely in the style of a high ischemic colic strictureplasty. And what we're able to do is we generate a pouch, allowing us to study the formation of strictures without causing a bowel obstruction. And as you can appreciate here in figure 2C, that we not only are able to successfully recapitulate creeping fat, but it's selectively in the animals that have undergone a colotomy and not in the sham treated, suggesting that it's a very localized effect. And if we look at the histology of our model, we see that it recapitulates key features that are present in human strictures. And these include excessive collagen deposition and increase in bowel wall thickness compared to the sham in our mouse model. And if we look at the extracellular matrix ultrastructure, that is the alignment and the thickness of some of the collagen fibers, using an algorithm that our lab has pioneered, we're able to see that the wounded bowel in the colotomy looks very different from the sham treated mice, as well as bowel that's adjacent to the colotomy site. In addition, we also noted the presence of adipocytes that had infiltrated into the bowel wall. And so next we asked the question, is it possible to lineage trace some of these adipocytes and thus be able to see if they're able to convert into fibroblasts? And so to do so, we utilize an adiponectin-Cre MTMG model. And so this model labels adipocytes green and any subsequent cells that are derived from an adipocyte green, and all other cells are red. And so what we observed was, as you can see in figure 4B, that some of these cells are green and have lost adipocyte markers, have acquired fibroblast markers, such as alpha SMA and PDGFR alpha, and that they also produce collagen and are responsive to GGF beta signaling. And so that suggests that these cells resemble fibroblasts. Next, we asked if creeping fat was necessary for the formation of intestinal strictures. And so to do so, we developed a model whereby we were able to selectively ablate fat and create a colotomy at the same time. And so you can see in the wild type that we have creeping fat, but in the ablated, we no longer have the creeping fat. And what's really interesting is that in the wild type, the bowel is very thick, but when we ablate the fat, the bowel returns to a much thinner state, suggesting that we're able to reverse some of the phenotype that is present in intestinal strictures just by ablating the fat. Next, we performed single-cell RNA-seq on mice that were either given a colotomy or sham treated. And here I'm just showing the colotomy data. And what you can appreciate are the presence of fibroblasts here. And then we've identified a particular cluster, cluster 2, which is enriched for ECM genes and genes that are responsible for collagen production. And so, and in addition, we've also identified fibroblast signature for this population. And we've done this in both the bowel and the mesentery. And we think that we have identified a particular population of profibrotic fibroblasts in both bowel and the mesentery. And then finally, I'll just point you to our last figure here, where we sorted green and red cells from our adiponectin Cre-MTMG model. And to remind you, the green cells are those cells derived from adipocytes. And what we've identified are cells that have the expression of fibroblast markers that start to resemble the canonical red fibroblasts that we would expect. And so it's interesting because it suggests that these adipocyte-derived cells start to resemble the standard fibroblasts that are present in the bowel and the mesentery. And so this suggests possibly that the mesentery may serve as a reservoir of fibroblasts, and that if we intend to treat intestinal strictures pharmacologically, we may also wish to consider the fibroblasts that come from the mesentery. Thank you so much for your attention.